First, I would like to thank the organizing committee to give me the opportunity to share with you today our results about the use of the lactic acid fermentation as means of nutritional improvement of Triticum, Turanicum, Bran. So I will start giving a brief introduction of the milling process. As most of you know, the milling process has the main objective to separate the endosperm from the other outer layers of the caryopsis, and while the endosperm is used as the raw material for the production of the most bread, baked goods, the bread and the bran represent about 27% of cereal milling industry output, and they are mainly directed to the feed industry, feed formulation industry, although they preserve a very good nutritional quality. So the question is, why these products are not properly reused or, let's say, valorized? Well, it has mainly one reason. For example, when a bread is produced using a reasonable amount of bran, it could lead to several negative aspects, as a weak gluten structure, a bad flavor and taste of the final products, and finally there is also another factor, that is an increased content of anti-nutritive compounds like phytic acids. But recently, the application of innovative technology for the pre-treatment of these kind of products have been studied. As an example, the lactic acid fermentation is nothing new, because fermentation has always been used as in an infinite kind of transformed products, and today lactic acid bacteria are recognized for their probiotic effects, and most important, because they are generally recognized as safe. So the aim of the study was to analyze and evaluate the quality attribute changes of wet bran byproduct treated with lactic acid fermentation using a single and a pure lactobacillus strain. The workflow of the experiment starts hydrating the native bran and then sterilize it. Then the inoculum was made at 10 times 7 column-forming unit of lactic acid bacteria. In this case, we used Lactobacillus rhamnosus, specific strain, that is the 1473, and then the raw material was incubated during 48 hours at 37 degrees. As you can see, the red stars and the blue stars refer to pH and mass balance checks and microbiological analysis. And I would like to specify two things. That the first is the autoclaving step was made to ensure the microbiological safety of the bran, and the main objective of the autoclaving was to work in total absence of interferences. And the second, why we choose the Lactobacillus rhamnosus, this specific strain? Well, mainly two reasons, because it has recognized properties of growth and metabolic actions, and of course it's never been used in lactic acid fermentation processes of cereal milling byproducts. In fact, it was isolated from Parmigiano Reggiano cheese making product, and as you can imagine, we are pretty expert in Parma about cheese production and cheese consumption also. Consequently, the samples were analyzed for their total phenolic content and the antioxidant activity using 3 recognized assays. Then the phenolic acid profile was quantitatively determined using a UHPLC MSMS analysis technique, and the metabolite identification using a linear ion trap mass spectrometry. And since the phenolic acid and phenolic compound in these matrix are present in two forms, mainly the free and the bound forms, and hydrolysis was required. Then the phytic acids and the water extractable arabinoxylan were determined using spectrophotometer assays. The total count, the total viable bacteria were, and the bacterial spores were part of the microbiological analysis. Well, as I said before, the first step was to ensure the safety of the product. For doing this, the total viable bacteria and the spore-forming bacteria were quantified before and after the autoclaving step. The total absence, as you can see, of colonies confirmed that the autoclaving step worked well. Moreover, after the inoculum, we checked the pH during the fermentation, and a strong acidification of the matrix was found, as you can see here. Then, in the same way, the effective growth of the Lactobacillus rhamnosus was verified, and indicating that the fermentation finally has occurred. So, moving to the phenolic content, as I mentioned before, here I represented the bound and the free component of the native bran and the fermented bran, during 24 and 48 hours. And in general way, as you can see, an increasing of free phenolic content, total phenolic content, was found, while a significant decrease in the bound total phenolic content was found until 48 hours of fermentation. Another aspect that I want to emphasize is that the bound component is, in this kind of matrix, in particular in bran, in cereal bran, is always the preponderant one. Then, regarding the antioxidant activity, it was measured with 3 widely known assays, and also in this case the free antioxidant potential augmented and increased during the fermentation. This is very, very important because an increased antioxidant potential, in particular the free component, could lead to a protection against the oxidation, and so doing so, an improved shelf life could be noticed. Then here is the behavior of the bound antioxidant component, and that was the opposite. So this means that the metabolic processes of lactic acid bacteria were in action. Now I want to show you the total the phenolic acid profile determined with an HPLC MSMS technique. The yellow line stands for the ferulic acid, that is the preponderant phenolic acid found in bran, in cereal bran, and as you can see, history repeating. The free component increases while the bound component decreases. The same was for the synaptic acid, but more interesting is the case of the caffeic acid. In fact, a high free component of the caffeic acid was found after 24 hours of fermentation, but the bound component of caffeic acid was quantified below the low limit of quantification. So this indicates that lactic acid bacteria may be responsible for the lignin degradation and thus release the monomers, in this case the caffeic acids. Finally, the whole picture of the 5 principal phenolic acid quantified in the matrix, adding the paracumaric and the hydroxyic benzoic acids. Another technique that nowadays allows to collect a huge amount of information is the non-targeted approach. Using this approach, in fact, we could identify some metabolites that could be a biomarker, represent biomarkers of fermentation. In this special case, we found the amino acid degradation. As you can see, the structures of phenylalanine was found, and then that was easily metabolized during the fermentation, producing the phenylactic acid and the phenylacetic acids, and their hydroxyforms. And here, as an example, I leave you the steps required for the identification of these molecules. So we extracted the ion the mass to charge ratio 165 of the phenylactic acid. We found that in the fermented Bran and not in the native Bran. And then we compare the mass and mass third spectra with some references. For the same way, other amino acids metabolites were found, and these were very important because could lead to they are potentially and could contribute or have bioactivity. Then, talking about bioactive compounds, also microbial metabolites of phenolic acids were found. In this case, the dihydroferrulic acid and the hydrocyanapic acid were found both in free form, and that is not for bioactivity, but this compound can contribute to the overall organoleptic characteristics that we found, for example, lactic acid and citric acid compounds. So there are still two questions to answer. Lab, in this case, adds also the ability to degrade the phytic acid, the antinutritive compounds, where there are mainly two pathways for the phytic acid degradation, and they can act in synergy. So the endogenous and microbial enzymes are activated by low pH, that is naturally produced during fermentation. And in this case, in our case, we found a degradation of phytic acid that reached at 38%, almost 38%. And this is understandable because the degradation was only attributable to bacterial enzymes because of the autoclaving step. Finally, at very last, I would like to spend a few words about the effect of fermentation on the arabinoxylans. Arabinoxylans are very important constituents of the cell walls found in cereal grains, and but more important is that the soluble ones are recognized for their positive impact on baked productions and baked goods. In our case, the content of water extractable arabinoxylans increased about 3 times during the lactic acid fermentation. In conclusion, the metabolic processes of the Lactobacillus rhamnosus permit to release part of the bound component, increasing the antioxidant activity of the final product that could be relevant against oxidation processes. Then the identification of microbial metabolites in their free form is very important, because these compounds may have different absorption and metabolic characteristics which may have significance in their bioactivity, of course. And finally, these changes are likely contributing to the differences in technological, nutritional and sensory properties, and we could confirm that lactic acid fermentation represents a valuable, innovative technique that could be a potential of valorization of these byproducts. Finally, I would like to thank my lab colleagues, my tutors, and you for your kind attention.